So just I'm going to summarize the overall work which is being done in India on the different aspects of phytoplasma. Next, please. Is already Dr. Brahmat has introduced, no, so there is no need to, is really a unique, microorganism and unique microbial kingdom of obligate parasite nature. Is already discussed, some very peculiar properties that is different from bacteria. So, and it is still little progress is there from abroad to culture some of the candidates species, and but it is not it is universal culture media available for cultivating all the candidate species. So still it is a challenge before us, and there is a great challenge before phytoplasmologists to develop a suitable culture media to culture all the for detailed biological and other properties of the phytoplasma. Next, please. As taxonomy is concerned, is already discussed, belongs to class molecules and very close to tails, having more than 87-88% homology. Next. So far, on the basis of conjure region of 16 ribosomal gene, 34 ribosomal groups and 44 candidate species has been identified. And from India, very recently, a new candidate species has been added as CAP saccharide, which infects sugarcane, particularly 11 subgroup PAP. Next. So these all lists, and from India, we have out of these 34, we have 10 different groups from India, affecting 172 plant species. Next. Regarding diagnosis is concerned, again is a great challenge. Dr. Brahmat has already discussed that there is a still some symptoms are there which is very peculiar to phytoplasma, but it is not again because sometimes we can symptoms, and because some of the symptoms are very common to viruses and phytoplasmas. But besides this, we have at the same time we have some very peculiar symptoms, at least which can give an idea to further pursue for phytoplasma identification. I will discuss in detail. Before PCR invention, total identification of phytoplasma was based on simple compound microscope staining, two different gene staining and fluorescent scanning, that is ADAPI. Still, at this advanced stage, it is a very recommended practice where facilities are not available, electron microscopy and molecular biology. Still, we can rely on a staining of infected tissues by applying either of the DIN or DAPI stain to confirm the presence of phytoplasma in insect effluent tissues. Electron microscopy is well known. It is very after several phytoplasma species mud has already introduced. They are having pleomorphic nature, restricted to the phloem tissues of the cell, and it is very well identified in electron microscopy. Serology, very little attempt, not like virus, because the cell wall is not there. So some antigenic membrane protein, they are purified from phytoplasma cell membrane, and polyclonal and monoclonal antibody has been developed and being utilized for large scale sampling. Still, serology has its own relevance because it is not possible to screen and index hundreds and thousands of samples for phytoplasma screening. So still, in for example, Iran, lime witch's broom, they have successfully developed very good high titer polyclonal antibody, which is being utilized for screening and index citrus varieties in Iran. Yeah, very little attempt has been done on sugarcane phytoplasma and coconut phytoplasma by applying some serological based properties, protein based identification. But still, in future, that must be in our target, because now because of climate change, we are realizing this incidence of phytoplasma more and more every year. So relevance of serology is very much concerned, and we must work on develop polyclonal antibodies, antibody kits, serological kits to identify phytoplasma in field-based samples. That is again very important. Like PCR, no doubt, after invention of PCR, now only came to understand more and more. Very reliable methods are available. Nested PC. Now we are not only sequencing as our ribosomal gene, we are using a number of multi-local genes. Now because now we have full genome sequence available. So now we have more and more idea of phytoplasma genome sequence, and now we can target different types of multi-locus genes. SEC-A, SEC-Y, TUF gene, GROAS gene, guidance B gene, DNA B gene. Number of multi-locus genes are being used for very finer classification of phytoplasma up to subgroup level. So now it is well in place, and mostly we have been utilizing in India for characterization of phytoplasma from a plant as well as from insect tissues. So it is this is the most reliable and authentic way. And once you have sequence available, then you can compare, blast, and get an idea of phytoplasma identity. Besides the recent, abroad, they have developed isothermal assays, for identification of phytoplasma. So this is again very friendly. Without a master cycler, we can develop reaction within a day, and this is very friendly. We can develop more and more samples, analyze samples with the help of either LAMP or RPSA. Next, please. As I told you, some of the symptoms are very important. Like little leaf, phyllodes, witch's broom, excessive shoot proliferation, flat stem, big bud. These are recent. In only 2-3 years back. First of all, we recorded these symptoms from Hyderabad, but within 2-3 years, it is so common, so emerging, that from at least 6-7 states we have been severe incidence of tomato big bud is there, and it is now 100% confirmed that it is because of Phytoplasma association. First group, second, and sixth group in different states. Phyllodes is again very well. So symptoms are again a very important criteria just to initiate the diagnosis process. So one working on Phytoplasma, little awareness and knowledge about symptoms, so that it will know immediately it can further move. You can move to further serology and molecular biology for further confirmation. Next, please. As I told you, now we can't say it is not culturable. The first thing is very recent, only 2-3 years back, Italian group of scientists in Bologna, they got very good success of cultivating some of the Phytoplasma on cultivated media by applying, this you can see soya broth, horse serum, this antibiotics and yeast extract. But again, it is not a standard. There is no standard formulation for any culture media for Phytoplasma. But it's a very good beginning, So now Indian scientists, we must look on it, and we should develop. We should work on suitable culture media for culturing different, because in India we have already reported 10 different groups of Phytoplasma. There is ample opportunity to work on culturing, because it's really, we can work, we can try different media, different combination, and definitely in future will very good, recommendation for universal media for cultivating these peculiar group of microorganisms. Next, please. As I told you, now more than 30 Phytoplasma draft genome sequences are available. So it is a very good development in Phytoplasma genomics. Now we have more and more information of full genome. We can have different down-regulated genes, up-regulated genes, and different factors. And from India, recently last year, our colleague Dr. Amit from Pune, they got success in getting full draft genome sequence of 2 of the species, which was not earlier available in any part of the world. That belongs to CAP-ORIG, that is 16SR11 group, and second one is Candidatus Phytoplasma cynodontis, that is 16SR14 group. These are very prevalent group, very dominant group in India, infecting a large number of, for example, CAP-ORIG. You can consider this is so important, infecting most of our important agriculture crops like sugarcane, coconut, erica nut, aisle palm, and other number of fruit species, and like Cepes, Synodontis is again very common to Gramineae family. Most of the wheat species and some of the cultivated species also. Next, please. Already know Dr. Bhaktamat has already introduced. We have a history of more than 150 years old, in India. When first of all, root wilt was discovered in 1874, followed by sandal spike, erica nut, yellow leaf, brinjal little leaves, sesame. So, you can see we have 150 years old history in India. But still, no, as suggested by earlier speakers, Dr. Krishna Kumar, we don't have any solid control measures to control any of these listed phytoplasma, which is still for the last 100 years we have been suffering, and it still is a greater challenge for all of us. So we must work on it to develop a very suitable, like insect vector. We must have knowledge of different insect vectors so we can formulate and suggest a very standard practice of control to minimize the spread of these phytoplasma diseases. Until date, I have very recently compiled all the phytoplasma reported diseases in India, and you can see there are 172 plant diseases so far reported belong to 10 different phytoplasma groups. Next. You can see the diversity. You won't find a plant where there is phytoplasma incidence is not reported. Starting from fruits, ornamentals, medicinal plants, vegetables, cereals, spices, pulses, oil crops. So this you can realize the importance. Earlier people they never considered phytoplasma is of economic concern, but now you can realize not in India, all over the world, it's becoming a very emerging and repeating phytoplasma diseases, which are really responsible for the very serious losses in all of our crops. So that is again a serious concern, and we, as suggested by all of earlier previous, we must have a coordination program, collaboration program, all the plant pathologists, microbiologists, entomologists, and biotechnologists to come together on a platform and to focus the major problem of the challenges of phytoplasma diseases in India. Next, please. You just know anybody can go through. It is already a recent publication. You can find all the this development of phytoplasma research in India, very recently published in Indian Phytoplasma. Next, please. Now come to, I will highlight some because time is less. We can't cover all the only I will focus on the major phytoplasma diseases. Sugarcane is again very important. There are two different types of symptoms, but phytoplasma is one, but this physiological expression is different. Sometimes we get white leaves, mostly in other Asian countries. In Thailand, Vietnam, Sri Lanka is a very common disease. However, in India, we mostly get grassy shoot disease. But at the same time, we do get chlorosis, but not always. So these are and that crop affects major the ratoon crops. And losses are reported every year, especially some of Tamil Nadu, Maharashtra, and western part of Uttar Pradesh. It is a very serious disease. And there is no control. Only there is no 100% resistant varieties. Only we are breeding tolerant varieties to come over this problem in India for the last 20-30 years. So now it is again a serious challenge, and now sugarcane breeders now they are planning to breed varieties again this particular disease. Next, please. You can see distribution all over India. Even in north part of India, everywhere recently, 2-3 years back, we surveyed all the northeast region and we recorded this particular disease. So it is really a problem all over India. So, but we have very little information available for its spread, insect spread. Hardly 2-3 leafhopper species have been identified. Next, please. See, just we discussed, there are two major these groups associated, 11B and 11F. And recently, Amit Yadav group, they have described as a new species for this 11th F, very close to CAP synodontis. And other sugarcane yellow leaf disease, first B group is reported associated. Next, please. No, so far, as I told you, these are the some species. So far, we have reported 2 natural vector, Cofana and Majus portico as a natural vector, and all they are Datos vulgaris from Lucknow, Indian Institute of Sugarcane, Lucknow, and Res exignatus and Pyrilla they have reported as putative vector. But as Dr. Braktamat had said, no, we have to work more and more to prove whether they are really true natural vectors of sugarcane or not. Next, please. Second important disease of sugarcane is the yellow leaf disease. It discovered in 1990s from Hawaii, earlier reported to be caused by sugarcane yellow leaf virus, that is the luteovirus. But recently, 4 or 5 years back, we reported Phytoplasma association. You can see, earlier we thought it may be because of physiological disorder. But when we tried, when we attempted in some of the affected field, from UP, all major sugarcane growing states of India. And now it is a major concern, not sugarcane grassy shoot. It is again becoming a very serious problem in comparison to grassy shoot, and we got association of first B group. And this already has been reported earlier from Egypt and Brazil. So it is again a big challenge for sugarcane growers. And because of this, we are each losing at least 5 to 10% losses because of this yellowing yet. And harvesting at least after 5-6 months of age, we'll get major this yellowing of symptoms. Next, please.